It's interesting to give the last lecture on blood pH because blood pH is a topic that brings together sort of a confluence of topics that go all the way back to the very first lessons at the very, very, very first start of this 30-some lectures ago. And also some things that were discussed in the middle, and then also fairly recent things. And we'll go ahead and throw in some chemistry there as well to really spice things up. Blood pH today. So let's begin with proteins and their shape. Of course, when we look at proteins and their molecular configuration in nature, in the natural world, in our bodies, the way that they actually exist, they tend to look like just a tangle of threads and craziness and not a lot of utility. But it turns out the shape of these proteins is actually really important. The shape of proteins gives them their function. The shape of the proteins allows them to have their functionality. Now, the name of this course is Anatomy and Physiology. Function equals physiology, and oftentimes students would say this is the most difficult part of this course, is to actually understand how things function. And it turns out that anytime you think about anything functioning properly, or when things go wrong with the way that cells function or tissues function or a disorder or so on, that there's always some kind of a misshapen protein, or a protein that's not produced in a large enough amount, for example, or a protein that's produced in too large of an amount behind the issue. So proteins, although we don't oftentimes say this throughout the course, they are essential for the function of your body. They're essential for the function of your body, and allowing them to have a particular shape that's everything. That's everything. So here we're going to explore, there's going to be a primary, secondary, tertiary, and quaternary structure. You can go back to that lecture if you like. But today we're going to be specifically discussing the secondary folding of the protein, which is based on an alpha helical shape or a beta sheet. And so let's just take a look at the alpha helix here. It kind of looks like a telephone cord. And in green, we're going to draw some dotted lines that indicates the way that these amino acids curl around each other, allows it to have this particular shape, which is mediated by a very weak bond called a hydrogen bond. A hydrogen bond is where you take a slightly negative charge from usually an oxygen and attract it to a slightly positive charge from a hydrogen ion or a hydrogen atom that's kind of further down the line of amino acids. And so it tends to take on this kink, and the beta sheets work the same way, but it's easiest to explore it in the alpha helix for sure. All right, so let me come back to that topic. The next question we want to answer is, what is pH? Well, pH is the negative log of the hydrogen ion concentration in a solution. And so we're going to draw three circles here. I'm going to be too high in hydrogen ions in one end, too low in the other end, and then somewhere right in the middle, kind of the Goldilocks situation, which is where our body exists in regards to how many hydrogen ions are appropriate. It may be most beneficial to start off with a number that is quite low. When we have hydrogen ions in a very low concentration, what we'll describe this as a 10 to the minus 13 is the concentration of hydrogen ions. Again, those brackets around the H plus means that we're discussing the concentration of something. So 10 to the minus 13 is the hydrogen ion concentration. And since pH is the negative log of the hydrogen ion concentration, this means that the pH of this particular solution is going to be 13. And to be clear, 10 to the minus 13 is an incredibly low number. It's like taking a decimal point and adding 13 zeros onto the decimal point and then adding a 1 at the end. It's a very, very low hydrogen ion concentration. Now, the reality is, of course, the way that you've been taught pH in the past is probably the idea that when you have something with a pH of 13 or very low, like 14 is as high as it can possibly go, this number can actually go. There's another substance called hydroxyls, which will exist in a higher concentration. It's an OH with a minus sign. And that makes the solution high in hydroxyls, but very, very low in hydrogen ions. Well, today's lecture, we're not discussing the hydroxyls. We're just going to be discussing the hydrogen ions in order to simplify this story. Let's turn to the other side of the story, where we have a very high hydrogen ion concentration. 10 to the minus 2 is incredibly high in concentration. It's 0.01. You know, it's just adding like basically moving the decimal point over by 2. It's a really high concentration, an incredibly high number of hydrogen ions, and therefore the pH is 2. High hydrogen ions will be a low pH number. Very low concentration of hydrogen ions will be a high pH number. And I do recognize that that's confusing, but hopefully having this visual there will help you to understand this concept a little bit better. And you may know that a pH of 7 is neutral, but it turns out our body's pH is about 7.4. And so our blood, our interstitial fluid, the lymphatics, all of the substances, all of the fluids in our body existed at a pH of about 7.4. So it's somewhere in the middle of 2 and 13, or 1 and 14, the lowest end and the highest end of the pH scale. Our blood exists pretty close to that central level. But you'll notice it's not 7. It's actually considerably higher than 7. I know that 7.4 doesn't sound very different from 7. But at the same time, every difference, even of like 0.1 in pH, makes a big difference in the ability of proteins to do their jobs. So again, let's put a visual on that idea. Let us, for example, represent the idea of a high hydrogen ion level. What if you dumped a bunch of hydrogen ions, which are basically protons with a really high electromagnetic pull, where you're going to be pulling in negative charges strongly? What if we dumped them into a protein? Well, what's going to happen is you're going to be breaking up the hydrogen bonds, which I already did say, it's not a weak bond. It's not like a covalent bond. These things aren't strongly attracted to each other. They're weakly attracted to each other. So if instead of an oxygen, which is partially negative, being attracted to a hydrogen atom down the line, which is partially positive, if all of a sudden you dump all of these strongly positive structures, these hydrogen ions, into that solution, that partially negative oxygen is going to be attracted for sure to all the hydrogen ions surrounding it, rather than the weakly positive hydrogen ion that exists as part of that protein. It breaks up the shape of the protein. Now let's just finish up the topic here. We're going to be describing anything with a pH of less than 7 as acidic, and anything with a pH of higher than 7 as alkaline or basic. So you'll notice that means that our blood, our body's fluids, are slightly on the alkaline scale. This is actually where most things in nature prefer to be. And why? Because when we have an alkaline solution, it tends to allow proteins to function the way that they are supposed to. If we have an acidic solution, which is the thing that we're most worried about in this lecture, to be clear, we're going to be centering our lecture basically on the hydrogen ions and the acid story, although the alkaline story is important too. The acid story is the big one. When we have a dumping of acids into our body and all of a sudden we lower the pH of our blood, for example, this could very quickly cause our proteins to denature, and they would no longer be shaped properly, and they would no longer be able to function properly because of that. Let us move on to our analysis. We're going to be looking into kind of the practical side of this entire story. First of all, our blood pH doesn't just exist at 7.4. It exists anywhere in a range of 7.35 to 7.45. Any number higher than 7.45 is considered alkalosis, an alkaline solution, and any number below 7.35 is considered acidosis. Your blood is becoming acidic, which again, the acidity is usually the problem that we're the most worried about. So if you are able to take a reading, a real-time reading of a patient's blood, you can tell whether they are at regular levels or whether they are undergoing acidosis or whether they are undergoing alkalosis, which in certain conditions, either of these situations may occur. Now there's a number of phenomena in place inside of your body that allow you to reestablish or to maintain your blood pH between 7.35 and 7.45. But the biggie, the one that's most important, and it kind of does incorporate lots of other things going on, is the bicarbonate buffer system. So let's go ahead and write it out. CO2 plus H2O. You'll notice that the arrows are doubled. We have double-sided arrows. Will convert into H2CO3. Another set of double-sided arrows. And then we have the conversion of H2CO3 into HCO3 minus and H plus. Now, I'm going to go ahead and I'm going to label everything in this bicarbonate buffer system, but I wanted to label the strong acid right away. H plus is a strong acid. I want to be clear about this. I'm labeling it in red every place that we see it, because we want to be clear that what we're looking at here is basically a single proton. It really pulls away bonds. You want to be careful about not allowing this equation to pull to the right so that you start to have large numbers of hydrogen ions. And so you want to kind of pull things to the left. I don't know if that makes sense. This is the way that it was taught to me. It's hard to, I don't know. I really don't know if it helps or hinders the understanding. But the basic idea is that you don't want to have an influx of strong acids. You don't want to have an influx of hydrogen ions, because this is acidity. So a lot of what's happening in this bicarbonate buffer system is basically trying to eat up acids that are produced by your body on a regular basis. And by the way, that's natural. You're always producing acids through basic metabolic functions. You're always producing some kind of acids, phosphoric acids or uric acids or lactic acids, famously by the muscles, for example. When we metabolize fats and sugars in our cells, the cells will put out metabolic waste products, which are almost always acid-based. Ketone bodies put out by the metabolism of fat. That's something that we've brought up before. So, yeah, we're always just worried about high levels of hydrogen ions, and we basically can't have it. It starts to bring our pH below levels of comfort, and the bicarbonate buffer system is about trying to fix that problem. All right, so let's go back and label everything. I think the first thing I want to point out is these unusual double arrows, and they can be written different ways, but this is the way that I've learned it. This is the way that I write it. So a reversible arrow is an arrow that indicates that an equation can go from the left to the right or from the right to the left. It can go either way. It is reversible. It is reversible. So in this case, we have this carbon dioxide plus water can be converted into this weak acid called carbonic acid. Carbonic acid is a weak acid. And then there's another, we're not going to label this, but I'll go ahead and say that also we have reversibility of the carbonic acid being pulled apart or being broken down into the last part of this, which is the HCO3 minus and the hydrogen ion. So that also can go to the right or to the left. So whenever you see a reversible arrow, whenever you see a double-sided arrow, it means reversibility. The equation can go either way. Now, let's discuss this carbonic acid. We call it a weak acid because when you place a weak acid into water, for example, well, it can go reversibly and incompletely either way. Unlike the strong acid, which we're showing here, which is hydrochloric acid, HCl, you'll notice the arrow is one way only. HCl is pulled apart completely and irreversibly into a proton, hydrogen ion, and then chloride ions, which are not really as important in this story. I just want to be clear, sometimes students think that chloride ions play a big role. They do not. They basically are there to bond to the hydrogen ion, but in solution they are immediately pulled apart and they do not go back the other way. They do not come back together. You have this hydrogen ion which is hanging out and just causing pure acidity, pure chaos. So yes, HCl is a strong acid, and the arrow that goes only one way indicates that this is an irreversible and complete reaction rather than reversible and incomplete. Okay, lastly, oh, what a golden structure. The bicarbonate ion is so important because it is the bicarbonate ion that allows us to basically eat up acidity in our body. We go to a lot of trouble to produce bicarbonate ions. The bicarbonate ion is a weak base. You notice it has a negative charge. It is sometimes written as NaHCO3. In that case, it would not have the negative charge on there. But I think it might be helpful to consider that, again, we're centering our understanding of this lesson around the hydrogen ion. And notice at the very end of this equation, we have this strong acid, this hydrogen ion. When the bicarbonate ion combines together with it, we can push that to the left and we can create a weak acid, carbonic acid, which does not contribute very much to the pH balance of the body. So this is why the bicarbonate ion is so important. As I said, we're constantly battling against the acidity in our body, and so the bicarbonate ion helps us to mitigate that problem. So that's the importance there. Right, let's turn our attention to the carbon dioxide then. It makes sense, although you wouldn't think of it right away probably, but it makes sense that the respiratory system is the primary system in charge of ensuring that carbon dioxide levels remain as they should be. About 200 milliliters of carbon dioxide are produced by the cells of our body at rest every minute, and even more, of course, when we're exercising and our muscles are working hard and you're producing lots of carbon dioxide as a result of aerobic respiration. And all of that carbon dioxide really needs to go. You cannot have high levels of carbon dioxide in your blood because that leads to acidosis. All this entire equation is pushed to the right, you see. High levels of carbon dioxide lead to high levels of carbonic acid, which in turn leads to strong acids being dumped into your blood. Like it's pushing. So what we need to do is we always need to make sure that we're getting rid of our carbon dioxide as soon as possible, and that is when we control our respiratory rate through our lungs, through our respiratory system. So we maintain our blood pH, our carbon dioxide levels in the blood at, well, you may recall that when blood is in the arteries and the systemic arterial system, making its way out to the tissues of the body, that the partial pressure of carbon dioxide at that level is about 40 millimeters of mercury. And as the blood passes through the tissues, it picks up some carbon dioxide that was left off by the cells in the tissues that are metabolizing, and the carbon dioxide level goes up slightly to about 45. So it turns out that healthy levels of carbon dioxide in the blood are between 35 to 45 millimeters of mercury, and your respiratory system goes to a lot of trouble to ensure that it is maintained so. Because as soon as you start to have higher levels than 45 millimeters of mercury, you might be undergoing respiratory acidosis. Basically, you're acidifying your blood because perhaps your respiratory system is not working very well. And by the way, this is maybe the most common issue health-wise that healthcare practitioners would have to worry about in regards to blood pH. A person's lungs are not working as well as they should, maybe emphysema, a different COPD disorder, something's going on, maybe COVID related, something, maybe some kind of other virus, pneumonia. You're not exchanging things as you should, and therefore you're trapping carbon dioxide in your body. That means that the partial pressure of carbon dioxide goes too high, and this is called respiratory acidosis. But of course, the opposite thing can happen as well. Under certain circumstances, you can actually get rid of too much carbon dioxide. Your partial pressure of carbon dioxide goes below 35. This would be called respiratory alkalosis. And one example of how this might occur is if a person is hyperventilating, they're breathing in and out, perhaps at a fast rate, but mostly at a very deep rate. Hyperventilation is like oftentimes because of a panic attack or certain pain situations, for example. In those cases, what's going to happen is you're going to be getting rid of too much carbon dioxide, and you might end up with respiratory alkalosis because your carbon dioxide levels are actually going too low. Well, the first situation we discussed would be considered when too much carbon dioxide is trapped in your blood is considered hypercapnia. And the other situation where you do not have enough carbon dioxide is called hypocapnia, these are terms that relate simply to the level of carbon dioxide in the blood and don't specifically infer acidosis or alkalosis. But we're tying them, of course, to that because of this lesson today. Well, going a little bit further, how does the respiratory system fix each problem? Well, and the respiratory system can work within 30 seconds to a minute to ameliorate whatever problem is occurring here, as long as everything's working properly in the lungs, which of course that's a big if, because oftentimes it's the lungs dysfunction that actually cause these problems. But if everything's functioning okay and you are undergoing hypercapnia, then you can simply raise your respiratory rate in order to get rid of more carbon dioxide. And the opposite side of the story, if you have hypocapnia and your carbon dioxide levels are too low, you actually want to lower your respiratory rate. You want to signal to those respiratory centers in the brainstem to lower your respiratory rate in order to trap back more carbon dioxide. So spend a little bit of time thinking about this. I think the hypercapnia and raising the respiratory rate, that's easier to picture exactly how that works than the opposite problem. I think it's difficult to actually engage with the lesson of hypocapnia because it's just a little bit odd thinking about lowering your respiratory rate, thinking about too little carbon dioxide trapped in the blood. It's just opposite of the way that we usually think of things. But I will say also that in either case, if you're either having too much carbon dioxide trapped in your blood, or too little, your body oftentimes will shut down. You'll get like the tunnel vision going on in order to allow your respiratory center to just take over. For example, the hypocapnia story, if you're having hyperventilation and because of a panic attack, for example, sometimes people pass out in those situations because the brainstem needs to just take over the respiratory rate for a little bit and improve things before the person can come back to consciousness. Right, let's take a look at that magical bicarbonate ion that fixes a lot of problems with acidosis. Well, it turns out that these kind of problems take a little bit longer to fix because we need to use the urinary system, we need to use the kidneys, and the kidneys, remarkable as they are, this is another amazing thing that they do. They maintain your bicarbonate ion levels at about 22 to 26 milliequivalents per liter. Small m, big E, small q per liter. And that's just a measurement of the concentration of these guys. So these are considered alkaline structures. They're negatively charged. They are alkaline. It is a weak base, but it restores alkalinity into a system where perhaps our levels are, we tend toward the acidic. So it's really important to keep the bicarbonate ion levels at a pretty decent level. We maintain our carbonic acid level at only about 1 milliequivalent per liter, but our bicarbonate ion levels between 22 and 26. And the kidneys are actually able to produce bicarbonate ion through quite a tricky process, which we are not going into at this point, but we are going to be saying that the kidneys help to produce bicarbonate ion levels, especially when they are a little bit low, or when you have lots of acids in the system. So anything over 26, you'll notice that I've been using the color blue for alkalosis, and I've been using the color red for acidosis, so we'll be in keeping with that. Just to give you the visual, I think colorizing things is helpful. So anything over 26 milliequivalents per liter would be considered an alkaline situation. We call this metabolic alkalosis, where for some reason the kidneys are producing too many bicarbonate ions. Really doesn't happen. Maybe there's certain situations where I guess intense vomiting. I've tried to find particular situations where a person might undergo metabolic alkalosis, and that was one of the examples that I came up with. I guess because you're getting rid of so many stomach acids that the bicarbonate ion levels actually kind of stack up because there aren't as many acids to grab and get rid of. I think that these things are quite rare. But the other side of the equation is probably a little bit more common when you have too few bicarbonate ions and they go below 22 milliequivalents per liter. This would be considered metabolic acidosis. And in this case, for whatever reason, perhaps your kidneys are damaged, perhaps the cells that are specialized in producing your bicarbonate ions are not producing them for whatever reason, then you would be undergoing a condition called metabolic acidosis, where you simply don't have enough bicarbonate ions to mitigate the problems that your body's always dealing with all of the acids being dumped into the blood, into the body's fluids. So less than 22 milliequivalents per liter, metabolic acidosis. Higher than 26, metabolic alkalosis. When a patient comes to a clinic with a particular diagnosis, for example, they have some kind of a respiratory issue, some of these levels can be pretty predictable. So I wanted to just point out that there's 3 different numbers that we look at when we're trying to figure out if a person is undergoing acidosis, alkalosis, and which type of either is happening. So the first number that we look at, and try not to get tricked out by this, is simply the pH number itself. If the pH is between 7.35 and 7.45, there is no problem, or the problem has been completely rectified by combining together the functionality of the respiratory system, CO2 levels, and the urinary system, the bicarbonate ion levels. And sometimes they can fix each other, if you will. If there's something going wrong with the kidneys, then the respiratory system may be able to make adjustments to make up for the issues at that level. So there can be compensation that occurs where we bring ourselves back into the blood pH that's an appropriate level physiologically for the human body. But of course, if your number is a little bit too low, even if it's like 7.33, that's acidosis. And if your number is a little bit too high, like 7.47, that is alkalosis. And of course, the numbers in a clinic that clinicians would be dealing with, they might actually be quite low. We've seen numbers like 6.8, or we've seen numbers much higher than 7.45, for example. And it is in those cases that you really want to look at which system of the body is the problem. And it would be no surprise at all if you're treating a patient with a COPD disorder, you're going to be expecting them to probably have some kind of respiratory acidosis because their lungs are really struggling to function properly to get rid of carbon dioxide. But sometimes you can also look at your bicarbonate ion levels and you might actually be doing some kind of compensation, as I said. So you have to kind of look at all three numbers. You have to look at your blood pH number, you have to look at your carbon dioxide numbers, and then you have to look at your bicarbonate ion levels. And then these things will kind of tell a story about the patient that you're treating.